Flowering time is a fundamental trait in plants, determining their reproductive success and agricultural productivity. The timing of flowering must be synchronized with environmental conditions to optimize yields, especially in an era of climate change, where unpredictable weather patterns threaten crop stability. The genetic control of flowering time has long been a focal point of plant breeding efforts, allowing scientists to engineer crops that flower at optimal times, ensuring adaptability to different geographical regions, seasonal variations, and fluctuating climate conditions. Understanding the molecular mechanisms governing flowering time is crucial for sustainable agriculture, food security, and the development of climate-resilient crops. Advancements in genomics, molecular biology, and quantitative genetics have enabled researchers to identify key genes and regulatory networks involved in the flowering process. Studies on model plants like Arabidopsis thaliana have significantly contributed to unraveling the complex genetic pathways controlling floral initiation. These discoveries have been applied to staple crops such as rice, wheat, maize, and soybean, allowing for precise genetic modifications that enhance yield stability in diverse environments. The integration of genome editing technologies, marker-assisted selection, and transcriptomic analyses has further accelerated the breeding of adaptive crop varieties suited for specific ecological conditions. This documentary delves into the genetic mechanisms underlying flowering time regulation, explores its agricultural implications, and examines case studies illustrating how genetic modifications have contributed to crop adaptability worldwide. By understanding the intricate relationship between genes, environmental cues, and adaptive crop cycles, we can optimize agricultural practices to meet the demands of a growing global population. Flowering time is regulated by complex genetic networks that integrate environmental signals such as photoperiod, temperature, and endogenous hormonal cues. In plants, multiple pathways interact to control the transition from vegetative growth to reproductive development. The major regulatory pathways governing flowering time include the photoperiod pathway, vernalization pathway, Gibberellin pathway, and autonomous pathway. The photoperiod pathway is one of the most well-studied mechanisms controlling flowering time. Many plant species can be classified as short day, long day, or day neutral. The core molecular players in this pathway include the constans gene and flowering locus T. In long day plants like Arabidopsis thaliana, Colorado promotes FT expression under prolonged daylight conditions, inducing flowering through the activation of floral integrator genes. In cereal crops such as wheat and barley, Photoperiod-Sensitivity is controlled by the PPD genes, which modulate the response to daylight. For instance, variations in PPD-D1 in wheat determine whether a variety is photoperiod-sensitive or photoperiod-insensitive, influencing its suitability for different latitudes. Similarly, in rice, the HD1 and HD3 genes function analogously to CO and FT in Arabidopsis, regulating flowering based on day length. The vernalization pathway ensures that plants experience a prolonged cold period before initiating flowering, preventing premature blooming in winter. This pathway is particularly significant in temperate cereals like wheat and barley, which require vernalization for proper flowering. The flowering locus C, FLC gene in Arabidopsis, and its homologs in cereal crops act as major repressors of flowering. Exposure to cold epigenetically silences FLC, allowing plants to transition into the reproductive phase when favorable conditions return. The Gibberellin pathway plays a crucial role in promoting flowering under non-inductive conditions. Gibberellins, or GAs, are plant hormones that activate genes like leafy, LFY, and suppressor of overexpression of constans 1, SOC1, leading to floral induction. This pathway is particularly relevant in short-day plants like rice, where GA levels modulate flowering independent of photoperiodic control. The autonomous pathway involves internal regulatory mechanisms that control flowering time independent of external cues. This pathway includes genes such as FCA and FY, which function through RNA processing mechanisms to regulate FLC expression. The interplay between these pathways determines the precise timing of flowering, enabling plants to adapt to different environmental conditions. Case studies, genetic adaptations in major crops, rice engineering, photoperiod-insensitive varieties for global cultivation. Rice is a prime example of how genetic control of flowering time has revolutionized agriculture. Traditionally, many rice varieties exhibited strong photoperiod sensitivity, restricting their cultivation to specific latitudes. The discovery of HD1, a homologue of CO and Arabidopsis, and HD3A, similar to FT, provided insights into how flowering in rice is regulated by daylight duration. The Green Revolution witnessed the development of photoperiod-insensitive varieties, such as IR8, which enabled rice cultivation beyond its traditional growing regions. More recently, the identification of the GHD7 gene, a key repressor of flowering in rice, has allowed breeders to fine-tune flowering time for different environments. By modifying GHD7, researchers have developed rice varieties that flower early in short-season regions, while delaying flowering in areas with longer growing seasons, optimizing yield potential. Wheat, the role of vernalization and photoperiod sensitivity. Wheat is grown across a wide range of climates, from temperate regions requiring vernalization to warmer areas where spring wheat varieties dominate. The introduction of photoperiod-insensitive alleles, such as PPD-D1A, has significantly expanded the adaptability of wheat. This mutation shortens the vegetative phase, enabling early flowering and ensuring that crops escape late-season droughts in arid regions. By combining vernalization and photoperiod genes, breeders have developed high-yielding wheat varieties tailored to diverse agroclimatic zones. Maize originated as a tropical crop, requiring long growing seasons, but its expansion into temperate regions required significant modifications in flowering time regulation. The key genetic determinant of maize flowering time is the ZMCCT gene, which influences photoperiod sensitivity. Variations in ZMCCT allow maize to flower under shorter day lengths, facilitating its adaptation to higher latitudes. Soybean flowering time is controlled by a complex interplay of maturity genes, E1, E9, that respond to photoperiod and temperature. The E1 gene acts as a major repressor of flowering under long day conditions, delaying reproductive transition. The presence of recessive E1 alleles enables early flowering, making soybean cultivars suitable for higher latitudes. The discovery of J and E2 genes further refined soybean breeding strategies, allowing the development of early maturing varieties for short-season regions. These genetic modifications have played a crucial role in expanding soybean production beyond its traditional growing zones, ensuring a stable global supply. The advent of CRISPR-Cas9 and other genome editing technologies has opened new avenues for precise manipulation of flowering time genes. Researchers have successfully edited FT, FLC, and GHD7 genes to create crops with customized flowering behaviors, enhancing resilience to climate variability. The genetic control of flowering time is a cornerstone of modern crop breeding, enabling the development of adaptive varieties suited for diverse environments. By deciphering the molecular pathways governing flowering and implementing advanced breeding strategies, scientists have transformed agricultural landscapes, ensuring food security in an unpredictable climate. The continuous refinement of genetic approaches, coupled with emerging technologies, will pave the way for sustainable crop production in the 21st century and beyond.